So, the gram stain. So the gram-positive bacteria and gram-negative bacteria. Key steps. Well, there's lots of steps, but the ones that are most important, that they need to get the bacteria fixed onto the slide. They do that just by heating it, and they kind of weld themselves on. But let's put this back to basics. So we're going to add the crystal violet stain, and then we add iodine after that, and then we remove those stains, or we may remove those stains with a solvent such as ethanol. So add ethanol, and then some of those bacteria lose their colour during that process, and some keep that kind of purple. So crystal violets, that's our kind of purple colour. And then after that we add another, so some stay purple, and then after this some of them kind of essentially disappear, they don't keep their colour, and we add another stain called a counter stain to show us those. Add saffronin, and that is pink. So those are the key steps. And why is it important that scientists can correctly identify bacteria that cause infections? And this is, to give the right antibiotics. I think give the right antibiotics. You don't want to be giving them antibiotics that are not going to act against that type of bacteria. And some, particularly gram-negative bacteria, are very dangerous. So some gram-negative bacteria are very serious. Why do the types of bacteria stain differently? Well, the gram-positive have a really kind of thick layer. The cell wall contains peptidoglycans, or that murin that we just talked about on the other page. And they've got this kind of thick wall with a single layer of peptidoglycan. So positive thick wall of peptidoglycan, which is murin, and then that takes crystal violet. Whereas the negative, they've got the kind of standard membrane, but then they've also got kind of a thin peptidoglycan layer there, and then you've got a lipoprotein with a thin layer of lipoprotein and a second outer membrane made of lipopolysaccharide rather than phospholipids. So negative have a second layer lipopolysaccharide, and so that does not stain violet, but we'll take up the counter stain of the pink. And this is why they respond differently to antibiotics. So some antibiotics cannot get through these layers and get into the cell. Some antibiotics might disrupt the cell wall or disrupt DNA, for replication or protein synthesis inside the cell. But to do that, you've got to get in. So some antibiotics cannot get into the cell.